Good morning, everybody, and welcome to today's joint webinar hosted by Antibody Studios and Gator Bio. I'm Subodh Nimkar. I'm the Director of Marketing at Gator Bio. Today's webinar is split into two parts. Rich Wong, he will do a brief introduction to Gator Bio and our products, and then the main plenary speaker, Dr. Allison Tong, will present today's topic, which is computer-aided design and characterization of multispecific antibodies for COVID-19. Before we get started, I'll do some logistics. Today's webinar is being recorded, so all the attendees and registrants will get a link within 3 days after the webinar. Please type your questions in the Q&A box, and we will answer all the questions at the end of both the talks. Any questions that go unanswered because of time, we will be answering those by emails. Again, we are excited to have Dr. Allison Tong here. And without further ado, I would invite Rich Wong, the product manager for Gator Bio's BLI platform, to begin his presentation.
Yeah. Good morning, everyone. I am pleased to represent Gator Bio and introduce our company here today. And the first question, who we are, who the Gator Bio is. So our company was founded by Dr. Hang Tian and Robert Zhang, the same people who created the original BLI, the biolayer interferometry technology. And we are headquartered in Palo Alto, California. And we also have another sister company called ET Healthcare, which focuses on the in vitro diagnostic. And we are a well-funded company with more than 50 innovative granted patents to back us. And also, our sister company, their instrument called Pylon 3D, has been installed over 2,000 instrument units in Asia region. And as for our this life science division under our corporation, our Gator Bio, Gator, has been installed. Our installation base has been growing rapidly globally. And we are manufacturing and doing those research and development in Palo Alto, California here, with our team of the experienced scientists and engineering. So we have our solid foot in this level 3 market. So our mission and vision is very simple. We want to bring the analytical solution for a better, faster, and cost-efficient biologic development to improve human health. And as a front runner, we want to explore the molecular universe with this BLI to help the scientists and the researcher to solve the difficult life science and diagnostic problems. So then, when we're talking about the BLI, so what is the biolayer interferometry? So as we know, the interference pattern, we all know there is the interference pattern. So actually, biolayer interferometry is measuring the light wave interference. And you can imagine a partial white light hit the upper and the lower boundary at each face of the boundary, and then reflected, either enhancing or reducing the reflected light. So So this change and the change in the optical thickness on the surface results in the modulation of the interference pattern. So you can imagine if there are two waves in phase, then they can combine as a constructive interference. But when these two waves, they are 180 out of phase, then they can combine as a destructive interference. So that being said, by monitoring this change over the time allows us to observe the binding behavior of the target molecules. So this is a small video to introduce this the whole idea. So our founder, Dr. Hong Tan, he applied this idea to this glass-made probe. And you can imagine with the light source on top of this glass-made probe, those light will go through this probe and reflect it at each face of the boundary. And you can imagine if the molecule binds to the surface, which change this optical thickness will result in this interference pattern. So we can observe the combination of the interference pattern got right shift, so that we can observe this updrift, this binding event. And if another molecule binds to it, then we can observe another nanometer shift to observe this another binding event, this association. Vice versa, if this molecule falling off from the surface, then we can observe this shift back, and then we can observe this dissociation event. So this is how we measure the biomolecular interaction. So the next question is, so what is the Gator? So what can Gator do for you? So actually Gator is a tool to expand the biotherapeutic discovery, ranging from the early discovery down to manufacturing and QC, including the antibody titer, yes-no binding, epitope binding, binding kinetics, and quantitation. So with those applications, so we can measure all the different types of the biomolecules, starting from the antibody and protein interaction for immunotherapy, and then protein-protein interaction, and also the peptide. So with our advanced technology, we can also measure the small molecular interaction with a larger protein. And also, right now, we never stop our innovation. So we are exploring our gene therapy solution and also the lipid nanoparticles. So we focus on the three main applications: quantitation, kinetics, and epitope binding. For the quantitation, so what is the quantitation? Quantitation, it means that if the most commonly used are usually is the ELISA or HPLC to detect how much protein you produce. But with this BLI technology, it is very easy and fast to detect how much protein you produce by using our technology. And also another most popular applications on BLI is we can observe the real-time molecular interaction to measure the whole kinetic interaction. And also we can measure those competition for epitope binding. Yeah. So right now, what is the Gator solution? Our Gator solution can be divided into three parts: the instrument, and the consumable, and the software. So with the instrument currently on the market, we have two different models: the Gator Prime and the Gator Plus, with the A channel system with up to 8 spectrometers in parallel to measure the condition. And also we have this user-friendly, and we want to make this software to be accessible to all the users, and with our wide range of the biosensor and consumables. So right now we provide a full suite of the applications, including the protein A, protein G, protein L, and the human FC, mouse FC, anti-human fat. And also, one of the most popular or common one of the most common purification tag is this histidine tag. And we also provide the antibody-based anti-his probe, and also its alternative, the nickel NTA biosensors. So our nickel NTA provides a single state regeneration and neutralization, including the recharging state. So which means compared to the traditional BOI, you have to have another additional recharging state. But with our technology, you don't have to do it with our nickel NTA regeneration buffer and nickel NTA neutralization buffers. So also right now, we are also providing another a solution for this AAV, this gene therapy, including the AAVX probe and also the AAV9 probe to do this capsid determination. So what is our BOI performance? So compared to the traditional BOI, so you can see we improve the higher, we have a higher capacity to capture the more binding protein, so that which can give us a higher signal. So you can see this data, we can compare to the traditional BOI, we have a higher sensitivity and a higher signal over this very, very small molecule. And also, since we have this higher signal, we can provide a better signal to noise ratio to get a better separation and the concentration for these binding events. And we never stop our innovation way. So we provide this so-called flex assay, which is reusable the strap evident probes. So we call it as reactivation because it's a little bit different idea to the traditional regeneration. We remove the whole interaction, the whole sample and the whole ligand analyte on our surface, and then recode this sensor. So which means that this sensor is very low user environmental friendly because you don't need to throw it away with your strong interaction between the strap evidence and the biotin. So here, and it is a very good product for those screening purpose. So which means that you can use it up to several times. And then the best part is you can swap in between different binding pairs with our flex assay. The next one is I would like to introduce our AAV solution. So currently, this is the only ready-to-use AAV quantitation probe on the market, which can save significant time, money, and reducing those uncertainty because we Gator Bio manufacture this probe for you. So AAV probes also have the same BOI advantage. It's the cruise sample tolerance, so no sample dilution needed. And also the data is very, very high correlation to the most commonly used method on the market, which is the ELISA. Yeah. So compared to ELISA, our Gator, we can provide a very, very fast experiment with a fewer steps. So you can see there's no wash step needed, and you can just put your sample into the machine, and then you can walk away. And then after 30 minutes, you can grab your coffee and everything, then you can get your data. However, compared to ELISA, it takes up to almost 240 minutes to complete this whole process with a lot of manual and washing steps. So how our performance compared to the current market, the other competitors? As you can see, our Gator right now, our AAV probes can provide a dynamic range from 10 to the power of 9 up to 10 to the power of 14. And please stay tuned with us because we will be providing the more detail about this ultra wide dynamic range solution sometime next week in another webinar. And with our Gator, so right now it's a CXP compliance. So our Gator provides the 21 CFR Part 11 software and with the IQ/OQ to help the biopharma company to meet this GXP compliance environment. And thank you for your listening. And I am turning to Dr. Titong to present her study.
Thank you, Rich, for that introduction. And thank you for that brief overview, because I think that's going to help with parts of my talk today. And so before I begin, I'd just like to thank the audience out there listening for joining this webinar today. I'm so pleased to have the opportunity to share Antibody Studio's capacity for computer-aided design for multispecific antibodies using our first-in-class COVID-19 trispecific antibody, as well as how we use Gator BLI technology to characterize this trispecific antibody. I first want to spend a couple of slides to just give a brief overview on Antibody Studio, Inc. We are a biotech startup located in the San Francisco Bay Area. In specific, we are located in Hayward, California. And what we do is we develop novel and complex therapeutic antibodies for difficult to treat diseases. Essentially, how we do this is we apply computer-aided antibody design and use our expertise in antibody biology to design therapeutics with better clinical success. Our business model is based on 4 pillars. In the first pillar, we do license out our preclinical stage assets, and we've done so in the past, and we're currently working on more. In the second pillar, we also have partnership and service. On the partnership end, we co-develop therapeutic antibodies with using our patented platform. And this is the case in our COVID trispecific antibody. And we're partnering up with China Resources Biopharma to further develop this antibody. On the surface arm of this pillar, we have antibody optimization. And this antibody optimization service that we perform has led to over 70 successfully completed projects. In our third pillar, we have co-development, and usually we develop antibodies targeting new biomarkers with academic institutions. And in our last pillar, we do in-house discovery, where we employ the use of our different platforms, such as our multispecific, internalizing, or catalytic antibody platforms. Oh, back to the other slide, sorry. And in many cases, our in-house discovery and co-development does have crosstalk with the other pillars. For example, in-house discovery projects or co-development projects can be licensed out or lead to partnerships, to great partnerships in the future. Next slide, please. We like to show this slide to highlight how we harness artificial intelligence, or AI, to rapidly generate therapeutic antibodies. And our approach has several interconnected steps. Our expertise in antibody biology with the application of AI can additionally aid in target discovery, but also in the optimization or modification of antibodies, which we validate on the bench. We can optimize antibodies with respect to these attributes seen in this box, which include humanization and immunogenicity and so forth. In addition to that, we can introduce novel features into the existing antibodies, such as in the development of bispecific T-cell engagers, in which we can do regional replacement, as seen in this example. We can also design complex multi-specific antibodies for a variety of applications. Ultimately, we believe that a better designed antibody will result in a better designed clinical trial. Recently, we have incorporated AlphaFold AI software into our antibody toolkit, which has revolutionized the playing field as it can quickly pull information from multiple databases to predict the best antigen antibody model. From this predicted model, we can perform a number of biochemical and biophysical analyses that can identify potential issues that can affect antibody safety, efficacy, or manufacturability. And then we can optimize or fix these antibodies. For more information on how we optimize these antibodies, please check out Antibody Studio's review for optimization of therapeutic antibodies. We have a little QR code there that you can scan to get to it quicker. In addition to that, you can also visit our website at antibodystudio.com for more information in regards to our antibody optimizations, which we have brochures attached onto one of the service pages. When it gets down to our technology platforms, we can do antibody discovery using well-established methods as such seen here. Applying computer-aided design and our expertise in antibody biology takes antibody design to the next level with a focus on safety, efficacy, and manufacturability. Our robust and patented computer-aided antibody design platforms are the multispecific antibody platform, and it will be the focus of this talk for today. But we also have an internalizing and catalytic antibody platform. In my last company overview slide, I just want to point out that we have applied these computer-aided antibody designs in several in-house therapeutic antibody programs that have been indicated for cancers, infectious diseases, and CNS diseases. But in addition to this, our versatile platforms can be applied to other therapeutic systems such as mRNA delivery, ADCs, CAR, and AAV therapies, leading to limitless therapeutic possibilities. Hopefully, this was a good introduction into what we do at Antibody Studio. I'll now move into the multispecific technology platform in the context of our trispecific antibody. In response to COVID-19, we decided early on that we wanted to use our innovative technology platform to develop therapeutic antibodies against SARS-CoV-2, a highly transmissible zoonotic respiratory virus. While a majority of individuals will get mild symptoms when infected with this virus, a vulnerable group consisting of the elderly, immunosuppressed, or those with comorbidities are susceptible to severe infections that could potentially lead to death. In a great medical feat, COVID-19 vaccines eventually came about, but even up to now, many remained unvaccinated and vulnerable to infections throughout the world. Therefore, there was and there still is a need for COVID-19 therapeutics. And based on this, we applied our multi-specific antibody platform to develop a COVID-19 therapeutic for prophylaxis and treatment to aid in this major public health issue. But this isn't the first time we use our multi-specific technology. And these are just a flavor of some of the multi-specifics we've generated in the past. You can see here that we have previously generated a number of multi-specific antibodies with different formats and different components, including the VHHs from our in-house libraries, as well as our novel knob into hole formats for better heterodimer formation for these asymmetric multi-specific antibodies. Overall, we hope you can appreciate Antibody Studio's capacity in designing and developing these complex multispecifics. We also want to point out that some of the antibodies that we have designed have already made it to clinical trial stage, or very close to getting to that stage. And so our technology is quite robust. The discovery process for the COVID-19 antibody was pretty straightforward. We panned our naive and humanized in-house nanobody library, performed screening for the S protein binders that can also block S-ACE2 interaction. Then we defined epitope groups and applied computer-aided antibody design to help with the arrangement of the VHHs and the addition of the Fc fusion protein, and characterized the antibody in vitro and in vivo. And that's a very high-level overview of the discovery process. In the next slide, I'm going to get into a little bit more detail, but I won't spend too much time on this since I went over the high level. Generally, from our panning of the VHH library, we mainly use the binding and blocking ELISA, as you can see in the formats below, for the initial screening. Ultimately, this led to some great results for some VHH candidates. And we also further characterize these candidates using the Gator BLIK assay. If you take a closer look at these KD values, you find that the KDs are already relatively low for some of the VHH candidates. But when the VHHs are joined together, for example, in a multispecific format, the KD further decreases, indicating the synergistic effect of a multispecific compared to a monoclonal VHH-Fc. The other important aspect of this slide is to show the following mechanisms of action, seen here in the left panel. The trispecific binds the S protein, thereby blocking the virus from the ACE2 host receptor interaction. We also equip this antibody with a functional Fc, which can induce Fc-independent and also Fc-dependent responses, such as antibody-dependent cytotoxicity, complement-dependent cytotoxicity, as well as antibody-dependent cellular phagocytosis. In the next slide, I'm going to talk about how we wanted to design a potent antibody. And so we strategized that we needed to adjoin VHHs that targets different epitopes along the S protein of the SARS-CoV virus. Furthermore, with this design, the antibody could better withstand viral mutations that can potentially affect one of the epitopes, but not all, which we now know is a true issue. Here, we performed epitope binding assays on VHH candidates to distinguish epitope groups. We found that when we loaded the Gator in the first panel in figure A with 2AFC, followed by the association of 2AFC again, or two other VHHs, we see no competition with 3F, but competition with 2A and 1B. Interestingly, in figure B, when we load with 1BFC first, then the other VHHs, we see that 1B does not compete with 3F as seen before, but now it does compete against 2A, indicating that 2A may share some portion of the epitope, but not all. And it also indicates that 2A is a different epitope that may be wider and may overlap 1BFC. Explaining the differences in the this explains the differences in the competition profile between A and B figures. In addition, when we load in figure C, when we loaded 3F first, followed by the other VHHs, no competition is observed between 2AFC or 1BFC, which has been consistent between the binding assays, verifying that 3FC falls within its own epitope grouping. We also performed ELISAs to verify this grouping scheme, and the percent difference as a means to indicate the competition, which is in green highlight, or no competition, which is in red, were analyzed. And we were able to verify the BLI results that we had seen on the Gator, ultimately leading us to at least two major epitope groupings. But as I mentioned earlier, there's actually three distinct epitopes for 2A, 3F, and 1B, as we believe that 2A may share an epitope with 1B, but it is potentially an overlapping epitope. The last slide demonstrated that we discovered VHHs, each with different epitopes. Now, the next set of experiments show the trispecific arrangements of these VHHs against different epitopes can allow for better efficacy and manufacturability. As seen in the ELISA studies, the trispecifics show better ability to bind the S protein and bind the S-ACE2 interactions than the monoclonal cocktails of the VHHs. And you can see the difference between the trispecifics, which are the blue and the red line, versus the monoclonal cocktails, which is the others. The pseudovirus inhibition ELISA also confirms this trend of trispecifics outperforming the monoclonal cocktails. As you can see here, the trispecifics are the blue and the red line versus the monoclonal cocktail, which is the green. AI and antigen antibody modeling predicts the epitopes of the antibody to the S protein trimer, further aiding us with a logical arrangement of the VHHs within the antibody. Also, biophysical analysis showed that 3F1b2a has lower aggregation potential with the best thermal stability compared to the others, indicating that this format will have the best manufacturability, and therefore we designated this candidate as ABS-VIR-001. We next characterized the affinity of this lead trispecific antibody using Gator's BLI-K assay and found that it had high affinity for the wild-type S trimer protein. But as you all know, this pandemic showed the rise of new key variants, two of which are also shown here. Press enter, please. Here, with respect to the S mutants, the antibody affinity remained high for the most part, validating that our design and construction of the antibody was able to withstand mutations from key variants throughout the pandemic. But binding is just half the story. Does our antibody block these key variants, which is another important component? Based on the BLI-K assays, we were able to quickly develop and characterize ABS-VIR-001's blocking ability. And in the absence of trispecific antibody, the mutant S trimers seem to bind ACE2 better than the wild-type trimer. Biologically, this makes sense, as these variants became the dominant strain during their time frame. In the presence of the antibody in the ABS-VIR-001, we do see that complete or near complete blocking does occur, further demonstrating the efficacy and durability of ABS-VIR-001 throughout this pandemic again. Since nanobodies are known for their excellent thermal stability, we wanted to further demonstrate that. So this is a study where we performed a long duration heating for 45 degrees Celsius for 4 weeks. Each week we took a sample and used the BLI to measure affinity using the K assays. Each week you could see that the affinity was maintained even up to the 4th week. In parallel, we measured some biophysical melting points that also remained relatively consistent throughout each week. In addition to the great features of this antibody, we have demonstrated excellent thermal stability that will be useful in manufacturing, as well as global distribution. To wrap this story up, I just want to say that ABS-VIR-001 is a highly durable trispecific VHH designed with Antibody Studio's multi-specific technology platform. We were able to demonstrate the robustness of our platform as well as the real-world application for COVID-19. ABS-VIR-001 targets 3 different epitopes characterized using Gator's BLI. We were able to arrange the best orientations of the VHHs and equip the antibody with a functional FC using computer-aided antibody design to elicit multiple mechanisms of action, including ADCC seen here in the bottom panel. We also applied our antibody optimization by making sure we kept safety, efficacy, and manufacturability in mind. In all of our in vivo data, we saw some promising data, and we like to tell you that the in vivo data is also very good. As you can see here in the upper right panel, you can see that ABS-VIR-001 potently prevents infection when provided as a prophylactic 10 hours prior to virus challenge, which is the red dots, or significantly reduces viral load when provided as a treatment post-challenge, indicated by the green or purple dots. Importantly, these trispecific antibodies continue to demonstrate efficacy against many key variants owing to its multiple S protein epitope design and configuration. And this is a summary on the bottom table below of the binding and blocking abilities. Lastly, we showed that the nanobodies, including ours, do live up to the reputation of excellent thermal stability. which has great implications for antibody manufacturing and global distribution. With that, I'd like to direct you to the references for all this data, as well as the reference to another way our VHH COVID candidates contributed to COVID-19 research. All the QR codes are there for being able to access these easily, but you can also find these on our website. And with that, I'd like to thank everyone for your time and attention. A special thank you to the Antibody Studio team for their essential, dedicated, and hard work during the pandemic. Another special thank you to China Resources Biopharma for sharing our vision and believing in our potential in our multispecific antibody platform. And finally, I'd like to thank Gator for this opportunity to showcase our multispecific platform, as well as their BLI machine that greatly aided us in the characterization of our COVID-19 trispecific antibody.
Thanks, Allison,
Yep.
for excellent presentation. We have a few questions here. And again, for the audience, please continue typing your questions in the Q&A box. I'll start with the first question, and this is more about the time required to do this kind of project, and how much of it is how much of the time is spent on AI versus the actual bench work.
I can have Yue answer this question. She's our president of the company. Yue, are you there?
Yes, so I'm there. I think it's case by case, but for this tri-specific antibody, what we already have is an AI designed humanized llama VHH library. So we start panning from this library. So the panning process is like 2 weeks for 3 rounds. Then we got like about 100 of S protein binders, then check for SAC2 blocking. So from design to bench, it's like, I think 2 months, yeah. And then the one situation is for this kind of project, the biology is correspondingly simple. It's binding, blocking, then testing pseudovirus blocking. So it's very straightforward. But if the biology is more complicated, like if we make a PD-L1 TIGIT LAG3 tri-specific antibody, you need to build up a lot of cell-based assay to serve as a readout for safety efficacy balance, then the time will be per round. Yeah, so it's case by case. Yeah.
Okay, excellent. The next question is, what are the other techniques you used to characterize these antibodies, both bispecific and maybe trispecific?
I can answer this one, Yue, if you want.
Oh, sure, you can answer, yeah.
In addition to the BLI and the ELISA, we also did some epitope mapping analysis where we did transfections of cells and measured cell binding in the context of deletion mutants to completely define the epitope of where the antibodies bind. So that's in our paper. In addition to that, we also, we performed animal studies. So we outsourced this, and they did all the animal studies, but we provided the material and purified the material. We made the material, and so in addition to that, when making the material, we have to also verify the quality of the material. And so we perform a lot of biophysical and biochemical analyses on that material as well. And that's also in our publications. In addition to that, we did do some FC functional tests. We have ADCC data that I didn't really get into, but I wanted to just at least briefly mention. What else did we do? I think for the most part, that covers it. Yue, can you add any?
Yeah, I think we did the ADCC. We also did the pseudovirus neutralization assay.
Oh yeah, the pseudovirus. Yeah. Mm-hmm. The pseudovirus neutralization assay, yes.
All right. There's another question, and this is about complexity or the development challenges between bispecific and trispecific. Can you comment on the level of work required when you go from bispecific to trispecific?
Allison, I think I can answer this question. Actually, the answer is it's not easy. So bispecific for manufacturability, usually it's durable. For trispecific, for most VHH, if you just link them together to form a structure like this, there will be huge aggregation issue. So AB Studio developed like a methodology to predict the manufacturability of each VHH and the possibility to connect them together as the bispecific or trispecific, and by doing some calculation. So basically, we have a manufacturability score, which is analyzed by our licensed software, which is based on biophysical, and our in-house developed calculation methodology, which is based on biochemical, like ionic bond formation, hydrogen bond formation, hydrophobic interaction. So then for each given VHH on 3D level, we can give a score. If the score is less than 500, usually bispecific or trispecific antibody, the manufacturability is durable. But if the score is between 500 and 1,000, there is space to fix. If the score is higher than 1,000, then usually we will give up that VHH because there are too many features needs to be fixed at 3D level for manufacturability. So when I talk about manufacturability, I'm talking about aggregation resistance, thermal stability, and the overall structure stability. So which can be analyzed by those AI tools. Yeah.
Okay. The next question relates to the biophysical characterization. What are the challenges for characterization when it comes to bispecific and trispecific compared to, let's say, normal antibodies?
Yeah, I think there are several challenges. So first challenge is even not bispecific or trispecific. A lot of VHH as itself, as the developability is okay. When you fuse the VHH to FC, there is aggregation. Then on top of such a VHH FC fusion protein, if you add one more VHH, two more VHH, the aggregation problem will be more and more. So that's why we did some in-house study, and we try to optimize our AI tools so we can not only predict which VHH has better developability in order to assemble a bispecific or trispecific in the presence of FC domain, but also we can try to fix some sequence with developability issues. Yeah. So I think that is the uniqueness of AB Studio's technology platform.
Okay, excellent. Those were the questions that I have seen so far. I'll probably give another 10 seconds if there are any other questions from the audience. Again, you can type it in Q&A or in the chat box. I guess there are no questions at this time. Again, if you have any questions for either Gator Bio team or for Antibody Studios, please reach out to us. And as I mentioned in the beginning, we will make the recording of this webinar available to everybody who registered in the next couple of days. And again, thank you very much, Allison, for a great talk, and Rich from Gator Bio. I see something in there is a question which is just came in: how to order the antibodies for bispecific? I don't know exactly what that question means. Can you please clarify when you say order the antibodies for bispecific?
Does that mean A, B, B, A, different order? How to arrange the order? Like, is that the question?
Yeah, I am not sure. Let's see whether we get more clarification. So yes, the question was yes. I think what you
Okay,
mentioned.
I see, I see. So first, based on biology, right? So if you make a T-cell engager, like the first domain is to bind to cancer antigen, the second domain is bind to CD3 on T-cell or something, right? So then there is like a proximity between the two targets, and then there is the order also for like there is also affinity issue. For example, the first domain, because it's well displayed, usually the affinity won't be affected. But the second domain, it's between like the first domain and FC, then the affinity will be started reduced. Also, the flexibility will be started reduced. In that situation, usually we will put the cancer binding domain as the first domain and the CD3 as the second domain, right? But then for other assembly, it's I think there are two determination factors. One is biology, like the mechanism of action. Second is the manufacturability. So sometimes like AB with FC has aggregation issue, but the BA with FC has no aggregation issue. So we will first analyze it in silico on 3D structure, then test on bench, and then move forward with like a living structure, then further optimize the sequence. Yeah.
There is a question, and this is for Rich. What is the throughput and runtime limit for Gator?
Yeah. So for current, our Gator Prime and Gator Plus, and when we're talking about a runtime, it comes to which application you're referring to. Let's talk about a quantitation. So for the normal quantitation, we have the normal our standardized default setting for the running time. And however, if we can, since we have a higher sensitivity, so actually we can shorten the time to reduce the assay time for this quantitation purpose. So on our current two Gator system, they are both the 8 peakers and connect to 8 different spectrometers. And also on our Gator Prime, you can use a 96 well plate to measure this whole interaction. On the Gator Plus, it can accommodate both the 96 well plate and 384 well plate. Both machines, you can also use so-called the probe holder, the max plate we designed for the probe holder. You can also pipette your sample or your buffer into the max plate, which compared to the traditional BOI, we improve the throughput on this 8-channel system. So for the quantitation, the normal quantitation assay time, if you're doing the 96 samples, so it will take you around an hour. But with the higher sensitivity protocol, then you can reduce the time down to 26 for 96 sample to do this quantitation.
Okay, thank you, Rich. Let's see whether there are any questions. Okay, so okay, there is one more. No, I think that's about what I see right now. So as there are no questions, I think I would again like to thank Dr. Allison, Titong, and Rich for excellent overview of Gator Bio and also Antibody Studio's approach to computer-aided design and development of bispecific and trispecifics. So thank you again for the audience for attending. I hope this was insightful for you, and we hope to see you again in our next webinar. We will be sending the links to this recording and so that you have access to it. Again, thank you, and we conclude this webinar.
Thank you.
Thank you.